Hi, my name's Josh Horton. I'm a second year PhD student at the Quadram Institute. Thanks for taking interest in my poster. So biofilms are a really important mode of life for bacteria, and it's a very complicated regulation process, moving from a motile to a sessile population, perhaps stuck to a surface or to each other. My project is essentially trying to figure out how biofilm formation occurs and the genes important for this process. Biofilms are really important because it protects the bacteria or the microbes from the external environment, and this can lead to tolerance to antibiotics, to sanitizers, which can be a nuisance in healthcare and the food industry. But because of this hardy characteristic, it can also be exploited for bioremediation. So my project is trying to figure out how maoP is involved in biofilm formation. Prior to my project, not much was known about this. It was discovered in a random mutagenesis screen for genes involved in biofilm formation. There's no homology to previously characterized proteins. The only thing that's really known about it is its importance in biofilm formation, but also that it's an RNA binding protein. The species of RNA is not really known at this point. So moving on to its impact in biofilm formation, we can measure this in multiple ways, but the two ways that I've gone for is measuring the production of a key protein called curly, which is important for E. coli and Salmonella biofilm. And this can be measured using Congo red dye, where if there's a lot of curly production, you have a red, dry and rough, such as in the wild type, whereas once you knock out maoP and you reduce curly production, it turns into more of a pink and smooth characteristic, which can then be complemented. This can also be measured using crystal violet dye, which measures the biofilm biomass overall, which deletion of maoP significantly reduces the production of biofilm in general. Moving forward, I wish to identify which RNA species maoP binds to, and this can be done by ClipSeq, which is cross-linking and immunoprecipitation followed by deep sequencing, essentially ChIP-seq but for RNA. And I've only done the early stages of this by making a tagged maoP, which I've shown that does not affect the biofilm formation in the strain once you add this C-terminal flag tag. So moving forward in the future, I wish to use ClipSeq and this now tagged maoP to identify which RNA species maoP binds to, and hopefully this will inform future hypotheses of biofilm formation in Enterobacteriaceae.